Hey everybody, welcome back. This is Sense Strand with a special edition. I got a brand new friend to help me out, debunk one of Dr. Sam Bailey's recent videos. Of course, you can't let go of the whole virus isolation thing. So to welcome Debunk the Funk with a Dr. Wilson, I got a special tribute.
Hey, Mr. Wilson!
Hey, Dr. Wilson, how are you doing today?
Hey, I'm good. How are you doing?
I was doing pretty fine, and then my blood pressure soared after I got a recent indication that our friend Sam Bailey has produced yet another video on viral isolation.
Yeah,
Yeah.
that you sent me that today, and that also just kind of ruined my day. But let's make it a little better by just you know
So
doing some face pumps. Yeah.
we'll go through minute by minute and just pause her where she gets some things wrong, and we'll criticize it right direct how it is. This is how peer review would go in a paper. We submit a paper and you go line by line, and
Yeah.
we criticize it. So I
Yeah.
think she gets on this rant about how voices are being excluded in science. And that's not true. I mean, if she had something that was worth presenting, it should go through the peer review process and then it gets peer reviewed. But what she wants is a double standard. What she wants is her videos to be presented in isolation without criticism, right? And then uses the entire, well, my videos are getting kicked off of the YouTube platform as a justification of why she's correct. But of course she's not. So
Yeah. Normally, she would have to subject her ideas to criticism by the scientific community and go through that whole process. But instead of doing that, she's just posting her videos online and convincing people who are not experts that she is correct.
And people who are already in their camp too. And also, I think I have to apologize because before, your videos also had a very stunning acceptance level. People who are scientists are on there like, this is great, these references check out. And then of course, I think I've introduced a lot of these Bailey supporters on there, and they get on your YouTube and thumb it down, and they don't make any actual points. And when they do make a point, a scientist will get on and usually like refute it. But
oh yeah,
I feel bad because I'm
no. Plenty of my videos have already been brigaded, I think,
right.
And I don't have the bandwidth to read all the comments, so I just let it go.
Right. Well, I appreciate your time, Dr. Wilson, on this one, because I do think she has quite an audience. She's like a silver rating on YouTube, and she's reaching a lot of people. So this is a big help. So all right, let's play this thing and see how we
go. And before we get too much into it, I want to say I haven't watched this video.
Oh yes,
but we were talking beforehand, and my prediction was that she is going to talk about isolation, but what she's really talking about is purification, which mean two different things in virology.
Yes.
And I have a feeling that she is going to say this virus hasn't been isolated when really she means it hasn't been purified, but it has been purified.
It has been purified and it has been isolated, but these are two things that you do for different purposes, correct?
Exactly.
Right.
Exactly.
So if you're going to gain an isolate, you need to do an isolation procedure. If you're going to purify the isolate so that you can image it or do some sort of extraction or manipulation on it, that's a different procedure for a different purpose. But she combines those two aspects in order to confuse the situation, because she's really a merchant of doubt. And she's a very good one, because as we'll see in this video that I've already seen, she's quoting actual scientists, but misquoting them, and even gives props to some scientists when they would not agree with what she's saying.
Oh, yeah.
All right, just raise your hand if you want me to pause this video and we can critique it.
One of the most misunderstood concepts of the current era seems to be that of virus isolation. Not a week goes by in which someone doesn't ask me to clarify what is going on, and I don't blame them, because even the scientific literature is a mess.
Is the scientific literature a mess on isolation?
No. I mean, see, the annoying thing is like, people like her will say things that have like a little kernel of truth. And
Right.
the only way you could possibly interpret like viral isolation as a mess is what Dr. Vincent Recchianello explains in his video, which she actually cites, which I don't know how she weasels around that. But in that video, he explains like uh, certain virologists might mean slightly different things when they say isolation, depending on which lab they trained in and how they use that vocabulary within their lab. But generally, when people write about it in literature, all the virologists know what they mean when they say isolate, and they have data to show what they mean by isolate. So it's all clarified, on a case-by-case basis
Right.
in every paper. So
So
I don't know what she means there.
when we say something like isolate, we mean a sample of infectious agent that's been sequestered from an individual, correct?
Mm-hmm. Yeah.
Right. And now with, I think she gets into it a little bit and then obscures it. I feel bad because I paused her on like a really bad moment, but it's going to happen.
Oh, well.
Dr. Bailey, I'm really sorry. I didn't mean to. But it's funny. So when we sequester this isolate, I mean, it comes from, say, working with bacteria or fungi. These are a little bit easier because they grow on a plate by themselves.
Mm-hmm.
And so when we have a single colony isolation, we know we have a single unit of a bacterial cell or a spore that we can say was an isolate. And for viruses, we have to do something special because they only grow inside of living cells. That's how viruses grow and replicate. But they still have this means of propagation that we can separate out and determine a single unit, which we then call an isolate, which we can pass along to other researchers and do experiments on, et cetera.
Mm-hmm. Yeah, and it's super easy to get a viral isolate from a cell culture. There are a couple ways labs do it. They can just take the liquid that the cells grow in. So that liquid is going to have cell metabolites. It might have some dead cells in it, but you spin all that down, all the heavy stuff falls to the bottom, and the liquid that doesn't get spun down with the cellular debris is going to have virus in it. That is an isolate. And you can further, refine that isolate by passing it through a filter, which is going to catch all large cellular debris and all bacteria and leave you with just virus. And that flow through the filter is going to remain infectious. So that is also an isolate. So slightly different ways of doing it, but it's all really simple to do, which is why so many labs have done it.
And the thing that I see Dr. Bailey missing out on here is, and she'll point at, is like, well, there's all this other stuff in there. So how are you even sure? And the point that I would get at is like, these viruses propagate. So you can do this again and again and again through cell cultures, and you come up with the same viral components.
Yeah. So. And also you can detect the virus in a patient, take a sample from them, and then propagate that virus in culture. Whereas if you had a control, a patient that you could not detect the virus in by all standards was uninfected, you take a sample from them and you wouldn't get that same virus in culture. So
right, that virus wouldn't come through, and also probably wouldn't match the test that you're running on it as well. But she will take this out of context, and we'll see further down the road. You'll see some pretty decent out of context stuff. I've on my previous videos on this channel, you'll see where I've selected specific images that she's taken completely out of context, and I believe she must know, because she literally took one table and removed the header that said clinical specificity and said these haven't been clinically specified. So I can't put it past her that she's probably doing this and knows what she's doing, unfortunately.
I think so. Yeah.
All right. But we're 16 seconds in. Let's get through it.
Let's see. Will look into some of the modern and historical aspects of the issue and why the terms isolation and isolate do not mean what many people think. The problem is when a virologist says we've isolated the virus, it implies a finality that needs no question. I'm not even sure if many virologists and scientists are aware of what they're doing anymore, and you're unlikely to hear any journalists call them up on the details. Although,
because why would a journalist call them up on the details? This is something that you publish through the science, and then your peers will say why you did this. And she actually has a really good example later on, which she obfuscates and are like, hey, you took these ultracellular images that don't seem to match up because they're not at a high enough resolution. Another scientist called them out on that. But why would a journalist come and say, you know, your isolation procedure is not correct? They don't know.
Yeah, I'm confused here. She's saying that virologists don't know what they're doing, but journalists know what virologists are doing. I don't.
That's the tag for this video.
I can't follow that logic.
Journalists are now better virologists than ever virologists wherever.
Yeah. No longer a need to do a PhD in virology. Just be a journalist.
Yeah.
sometimes.
Well, you isolate a virus by
He never comes back to this. I don't know what that's from. We'll have to find out what that's from, but I'm sure he had a competent answer after the second that he had to like take to phrase how he was going to say, take the supernate and spread it on some cells, get back out the virus that you're looking for.
Yeah, wow. That's probably some blatant cherry picking there. I'll have to find that original video, which I don't think she links in the description, because I did check out the description before we got on here. But I've heard David Baltimore speak, and he's still pretty coherent in his age, and he's a great scientist. So there's no way he doesn't know what a viral isolate is.
Yeah. God forbid anybody take a second to like think about what you're going to say before you say it. That would mean that you're good. Yeah.
Oh, and also it's funny because David Baltimore, he's a Nobel laureate, if I'm not mistaken. I'm pretty sure he is. And so it's weird to me when people like Sam Bailey talk about Kary Mullis, for example, and hold up his status as a Nobel laureate above all else, but then they kind of ignore any other credentialed people of Nobel laureate status, even, who might disagree.
Well, they weren't rebels who just abandoned science after their one thing.
Yeah. That was what Kary Mullis was. He became an AIDS denier.
Yeah.
So.
By the way, I loved your video. I totally missed it. I didn't even read his patent. I went through and read Mullis's like original papers. He was brilliant, of course, and he did suggest to use Thermus aquaticus for attack polymerase. But then all of that work was done after he left. Including the probing, which is a very important part of how you generate PCR results. For diagnostics.
Right. Yeah. He developed the initial original PCR, but qPCR is a very different variation of that. That has kind of different technology that is essential to that whole process, which Kary Mullis had nothing to do with. So.
Right. Yeah.
Yeah.
But, glowing raccoons. Remember
God,
that.
Yeah. I heard that story in college, and now his name is all over the internet, and I'm like, I remember that guy.
Just like he would have loved it that his name had gotten such a revolution off of being just so wrong about his own method. It's crazy.
Wild.
As a little introduction, we should quickly establish the definition of a virus. Essentially, it refers to a tiny infectious particle inside of which is some genetic material, which may be DNA or RNA. As I've discussed in previous videos, the purported SARS-CoV-2 PCR tests are supposed to react to RNA sequences specific to the virus. But before validating these tests for any use, it would need to be established that the RNA comes from inside the virus particle. That's because viruses only grow in cells, and cells already contain enormous amounts of genetic material. So first they have to be separated. Otherwise you won't know what's in your mixture. That's where virus isolation comes in, and you might be in for a surprise, as the language being used by the virologists is a little murky, to say the least.
Hold on.
Yeah.
Hold on.
Did you want to? Yeah. Go ahead.
Hold on.
Of course, that was.
So she's. So hold on. So okay, let's follow this logic. So she's saying that a PCR needs to be used on isolated virus
Because
there's not enough of it in the sample, and you have. Can you decipher what she's saying?
Yeah, I
turn it
believe
into
what she
something
is
coherent?
trying to say is that you can't prove that the RNA came from a virus because you didn't take it from inside the capsid, which is absolutely insane and preposterous because the viral sequences have nothing to do with cellular RNA that exists. You know, your
Yeah.
cellular RNA has no relationship whatsoever to viral RNA. And of this length too, of 29,000 base pairs. I mean, just 100 base pairs of viral RNA is not going to match your own cellular RNA.
Mm-hmm. Yeah.
But her contention is that, well, because you hadn't sequenced it apparently from a viral, from solely viral capsid, then you're not sure that it just doesn't devolve from your own RNA, because as she points out, mistaken Barbara McClintock's entire hypothesis, she thinks that de novo, your DNA and your RNA are going to make these just random sequences that are 29,000 base pairs long, which we know is not possible. It's not something that's going to happen.
Yeah, it's so we sequenced the human genome. I mean, almost actually, 20 years ago now.
Right.
Human Genome Project was finished, and or maybe just under 20 years. But I mean, we know what the human genome sequence is. We can compare other sequences to it pretty easily, and if nothing matches the human genome sequence, then you're not going to have any RNA that looks like a virus. But I think that what I kind of heard her say there was there's so little of this RNA that you're detecting, and it's that you can't be sure that it's from a virus.
Yeah.
And she might be including some of Barbara McClintock's work, which she talks about in other videos there, which she totally misrepresents. Another Nobel laureate that she just totally doesn't understand.
Definitely a hero of mine too.
Oh yeah, Barbara McClintock is great. But I mean the point of PCR is to be specific and then amplify the genetic material to a point where you can measure it. So yeah.
Well,
you all, those listening can tell. I mean, we're having a hard time just understanding what she's trying to say, let alone having it make sense with known scientific knowledge. I mean, it doesn't follow.
I mean, she's purposely being confusing, and she has jump cuts in there where she stitched some things together so that they sound like they make sense, but they don't make any sense whatsoever. The viral sequence is unique. That's the point of it, is that it is unique. And it doesn't matter how small it is, the amplification of that amount will produce the unique sequence. And that unique sequence through amplification cannot just be randomly acquired. Otherwise it won't be repeatable, first off, it can't happen, and second, it won't be repeatable, because you can't just rely on PCR error to generate an entire viral genome.
And I think maybe the other thing she might say in this little bit here is she's complained before about the size of the genome that the PCR amplifies, saying that that's not indicative of a virus. It's just a tiny portion of the genome. But a couple of things there. I mean, if you can amplify that specific piece of the genome from the virus, it is not a leap to infer that the rest of the genome is there, or else you would only because if the rest of the viral genome is not there, then it presumably already got chopped up into fragments by your immune system, and you wouldn't likely be able to get priming with your probes.
Right.
And also, that segment of the genome is actually pretty big when it comes to specificity. About 20 nucleotides of DNA or RNA can be entirely unique depending
Yes.
on the organ, depending on where in the genome you are. And that's what scientists pay attention to when they design PCRs. They make sure that they're targeting unique parts of the genome.
Right. And it gets back to basically what we call phylogeny, which is the relationship of organisms genetically. And I see this a lot in the comment section in your videos as well, is like, well, you haven't sequenced every single organism on Earth. So how do you know that this isn't, always was there? It's because we've been looking at these things and we know what a new virus looks like. Even if you haven't sequenced all of the organisms on the Earth, you sequenced a large enough proportion across all of the kingdoms of life to know that these sequences are related and are brand new. And just to get back to just to get back to your PCR example first, is like, right, I don't think people realize that there are 3 levels of complexity that go into a quantitative PCR. There's the forward primer, the reverse primer, and the probe. So a 20-mer, we call it 20-mer, so 20 bases on the forward side, a 20-mer on the reverse side, that has to amplify a specific length, which is usually about 70 to 120 base pairs long. And if those primers aren't together right next to each other, if they're off, then they won't amplify enough to make the RT-PCR reaction. And then that probe has to be set straight in the middle of that amplification in order for it to work, which is why you can run things out to 40, 50 cycles and still get negative. Yeah,
because even if you get some off-target effect on the reverse primer, for example, which people found, there's some weak off-target binding on human chromosome 8, I believe, for one of the reverse primers of the COVID PCR. But if the probe is over here and the reverse primer binds way over here, and the polymerase goes that way, it's never going to touch the probe. So you're never going to get any fluorescence from that. You have to have all 3 components lined up correctly.
At one, because one of the primers did match something close enough, so there's some slop that a primer will take that'll have like low efficiency amplification, but it's nowhere near anything that'll make a product. So that's back in. And actually, one of the things that I was looking at, the original like Drosten-Korman PCR, I think it used an RDP, RNA-dependent polymerase gene as a target that is very similar to SARS-CoV-1. And then people were pointing to that saying like, oh, look, it's not specific. And it's like, SARS-CoV-1 is not in circulation. If it's being amplified, it's far, and there's 3 genes that they're targeting.
Yeah, so, and I believe they even, they didn't have clinical samples of SARS-CoV-2 for that paper.
Right.
But they had, they were able to test whether or not it can also detect SARS-CoV-1. And if I remember right, they did not get amplification for SARS-CoV-1 with at least one of their primer sets.
With one of their primer sets they had. That's true. It was specific. And a lot of people dunk on that paper as not being perfect. And it's like, it was in January, right at the beginning of the pandemic. I expect RT-PCR targets to morph a little bit and become more specific as we got going on. But anything to throw doubt into the situation.
We can keep going now.
Oh yeah, right. No, we're so far off. Sorry, apologize. All right, back to isolation. About what she's gonna, she's pulling up dictionary items here. This is a pretty typical, Yeah, antis, people who are like trying to pull something over, they'll go straight to a dictionary definition instead of looking into what a scientific term is. So here we go.
So firstly, the word isolated derives from the Latin word insulatus, meaning to be made into an island.
Who cares?
Now let's consult the dictionary to see what these terms are usually understood to mean today. So recently, many people got to experience the first definition of isolation, although you may not have been officially called a prisoner. Anyway, isolation means the fact that something is separate and not connected to other things. So I think we could say that using this definition, we would expect that isolation of a microorganism or virus would mean that it has been separated from other biological material and we are left with just the object of interest. And if we look up the dictionary for isolate, we see that with regards to chemistry and bacteriology, the verb means to obtain a substance or microorganism in an uncombined or pure state. And with regards to the noun, it means a person, thing, or group that is set apart or isolated as for purposes of study. However, once you start searching for definitions of isolation with regards to viruses, you are unlikely to find such clear-cut terminology. In fact, some might say you are getting an introduction into a little secret of virology. And the year 2020 saw the issue become much more public as many more people became aware of the terms and techniques that virologists are using.
And it was working fine till you gang of snoopers came around.
Like we're trying to pull something over. What did you think of that? First off, right, there's some confusion in terms of,
Okay.
You're right. Yeah, go ahead.
I mean, well, first of all, she's appealing to the like anti-lockdown mindset, the which
You're
I
a
mean,
prisoner.
yeah, like I don't understand this idea. Like, you were always allowed to leave your house. You could go outside if you really wanted to. I don't understand the really strong language about this.
Well, if you don't believe the virus is even real, which she is either borderline on or is totally on, then entire world governments who can't get together on climate change, who can't get together on important issues on, I don't know, protecting the world from asteroids or space debris, but they can get together and decide that an entire virus is fake, spread around the entire world to make people stay inside and shut down economies. I don't get it.
Yeah, it's definitely appealing to just the conspiracy mindset, and I think she's treading that line on purpose to gain clout, gain followers.
It's clearly her.
Like we've said, she's kind of deliberately leaving things out of certain things, which tells me that she knows exactly what she's doing. And I don't like to think that about people. I like to give people the benefit of the doubt and say they probably genuinely believe these things, but it's hard to say that in her case. But with what she's just said about isolation, I mean, just like we were chatting before this, you can look up terms for words in dictionaries, and they don't necessarily mean what they mean in science or different scientific fields even. And you might, I mean, she's about to talk about Vincent Racaniello, and I don't know how she's going to weasel her way out of this, but Vincent kind of explains pretty clearly that, viral isolation does not have a clear-cut definition, but it has a general meaning that all virologists understand, which is just obtaining an infectious sample of virus that doesn't have any other, biological contaminants in it, meaning doesn't have any other viruses in the sample, no other bacteria in the sample.
Right, which you can do.
Yeah.
Yep, just through plate assays, culturing. I mean, this is 20, if not 40-year-old techniques of getting isolates. We do it in almost every organism you can think of, including in my work, we do it in plants, and we do it with like infiltrations, and it's very easy to demonstrate. Actually, we don't even have to. We can add them to particle grains and spray them on, and they'll infect. So
that's
yeah.
cool.
It is. It is. You just spray
Those
them
techniques
across
are
a
awesome.
little bit of damage, and they replicate without a doubt. They'll even carry genetic material if you want to do transformations and stuff. It's pretty cool. But yeah, here she's playing with the word. She's using the definition. She's abusing it because it was by applying it to bacteria, which you can isolate because they grow on their own, and then just totally ignoring and obfuscating the point that viruses are obligates, that they don't grow. They don't do anything. They're innate until they come in contact with a host. So you have to do this in order to isolate. And she's using that totally to her advantage to just kind of smear it and, give it a haze over the wording. But, it's
Right.
the CDC itself has isolates that you can request as a laboratory. So it's not that they don't exist. They totally exist. All right, let's see what she gets with.
I was gonna say, totally to that point of just confusing the fact that viruses need cells to grow. When you take liquid from a cell culture that is infected with a virus, in order to take your isolate sample, you're going to have cellular debris in that. You're going to have metabolites. That doesn't matter. It's still an isolate. As we'll get into, if you want to remove all those other things from that virus, you can purify it, which is really, straightforward to do. I've done the technique that you would use to purify virus. It's not hard to learn. And also, if you're worried about the cellular debris in that sample causing any disease or problems you might see, you can filter, or you can just take the same exact sample from an uninfected cell culture, and I can guarantee you're not going to get any infectious results by just taking used cell media and transferring it to another cell culture or an animal. You're just not going to get any viral effects.
Right. I don't know why they would think what they call like cell toxicity. What they just mean is I'm assuming like waste product from cells to propagate and cause disease again and again and again after petri dish after petri dish. That's just not going to happen. It only will happen in a biotic agent that causes infectious disease,
right?
Like a virus that propagates.
Yes.
So, all right. Let's see how this is going to disappoint you. So let's see.
Oh man.
I'm going to quote Vincent Racaniello, Earth's virology professor, a few times to illustrate some of the problems. Now, my opinions about fundamental principles of what are termed viruses and how they relate to disease differ sharply from Professor Racaniello, as he is part of the orthodox school, but I find him more internally consistent than many virologists. Despite the fundamental differences in belief, you could even say I have a soft spot for him, and if you want to learn about establishment virology theories, then he's worth checking out. However, as we're about to see, even the professor seems to start well, but then gets loose with the language when talking about viral isolates. So what does he say about the terminology in current use? Many people are thinking about viruses, and so they're using terms they may not be familiar with, like isolate, variant, and strain. Racaniello points out how most of the time in the field of virology, doctors and scientists simply parrot the terms that those who influence them are using, which may or may not have any consistency. He states, unfortunately, there is no universally accepted definitions for the terms strain, variant, and isolate in the virology community, and most virologists simply copy the usage of terms from others. Interestingly, Racaniello goes on to say that even in the 4th edition of his textbook, Principles of Virology, he and his co-authors had neglected to clarify these terms, something they will perhaps look to do in a future edition. I think you can already see that there is a major problem here. We expect scientific language to be very precise. For example, in the field of aerospace engineering, you wouldn't want the scientists to have loose definitions of important terms.
Yep.
All right. Okay, so she just completely
Ignored.
kind of glossed over what Vincent actually says in his video. It sounds to me like she's trying to paint this picture of, virologists just don't think for themselves and they copy others or parrot each other. That's not what happens.
And I believe what Vincenelli was saying is that you've imported these terms from other organisms that make them a little bit less, a little bit more clear cut. So, but they're useful terms. They're incredibly useful terms. An isolate is an infectious reagent that you've sequestered from an individual who is infected.
Right. Yeah. And there are a couple different ways of obtaining that isolate, which, I think lends to what Vincent said about depending on whose lab you worked in, you might use that term differently and so on and so forth. But it's all generally the same thing. And yeah, the not every lab is going to use all the terminology that like variant and strain. They're not going to necessarily distinguish between those, but in the context of a pandemic, then those definitions kind of change. So for example, if I were in my graduate lab and we were working with yeast, every single yeast that I have on a plate, it is a strain. But if it does, even if I make a mutation to a yeast,
right,
and it has no effect, it just looks exactly like wild type, behaves exactly like wild type, it's still, we still call it a strain in the lab. But in the realm of this pandemic and virology, if you find a virus that is mutated slightly from the original Wuhan strain, but it behaves no differently, it has no new biological functions, then it's a variant, not a strain. So there's nuance. There are little caveats to all these definitions depending on what field and what situation you are. And as long as, the scientists working directly with those terms can explain them properly within context, then it's fine. Because at the end of the day, with language, as long as you understand what the other person is talking about, then it doesn't so much matter the exact words that you use.
Yes.
But Vincent, being a professor, he gets really picky about how certain terms are used, and it irritates him. So he talks in a way that you can cherry pick, the way Sam Bailey did, to say, oh,
it's slop.
Everything is sloppy, when that's not the case.
No, it's not the case at all. And he knows exactly what it means in the literature when you say variant, right?
Yeah.
And the variants become important because you don't necessarily know the moment that you've obtained a variant if it's going to behave differently. That takes a lot of monitoring.
Yes.
So
yeah.
So each of these terms have become, at least commonplace, and they are, the purpose of them is to keep track of what's going on. There are thousands and thousands of variants because this thing is evolving so fast. And also it's biology. Things tend not to be cut and dry like they are in mechanical engineering. Right.
Yeah. And I'll give her some credit for saying saying that Vincent's main views disagree with her. But then she totally backpedals and says, but he's from the orthodox.
He's from the orthodox. Exactly.
What? Why talk like that?
I don't know. It's a shit because she wouldn't directly she takes his words, cherry picked them out of context, but wouldn't go to the viruses, isolates, variants, and strains, which we can listen to briefly just to see if he comes up with it really quick. I mean, it's 25 minutes long. You should everyone who's listening, go watch that video.
Go check
that video is going to actually tell you something. Okay.
Yeah, he only spends the first eight or so minutes talking about isolates.
Right.
But during that time, he shows a picture of all the variants, or maybe it's when he talks about variants, I can't remember. But at some point, he shows a picture of all the variants. Oh yeah, I think it's next strain right there.
Yeah, right here. Just play it quick and let's see how it goes.
Or now I clicked on one of these blue dots downstream of it in the phylogenetic tree. And you can see here it's got an isolate name, Bulgaria 15 2020. That's great. We know it came from Bulgaria. It's given an isolate number 15 in 2020. And in fact, May 13th specifically. And you can see it has these genome changes compared to the Wuhan virus.
Right. Those genomic changes compared to the virus. So that's a variant they're calling, and they're tracking all of those.
Yeah. So, and they've identified all of these variants because they've been isolated. All these little points are viral isolates. So if she had, talked about this part of the video, her argument completely falls apart, because there are at least this many labs around the world who have isolated virus, sequenced it, and found it to differ from the original Wuhan strain by just a couple amino acids. And there's no way you're going to get that unless there is a singular species, so to speak, of virus that is propagating around the world.
Right.
You're not going to get that if it's cellular debris. You're not going to get that if it's exosomes. You're only going to get that if it's a propagating virus.
Right. And each of those labs that are doing those isolations, Sam Bailey's point is always like, these are just remnants of errors in PCR. Well, then they're not going to group in a clade like this. They're not going to clade out like that. It's going to be random. They'll be random. They won't group in clades. So
yeah, and they sequence the whole genome of the virus in order to see these changes. So I don't know how you get random errors in PCR that amount to only a couple changes in amino acid sequence across thousands of isolates.
That's too consequential to be an error that happens all of the time across all of these laboratories. But that's her point.
She's just how did she?
Listen, Debunk. I idolize Dr. Bailey and all of her points. So let me cherry pick what she says and take it completely out of context.
Oh God.
All right.
Acceleration and center of gravity. In the past year, we have seen numerous papers reporting that they had isolated a certain famous virus. Subsequently, fact checkers and mainstream media acolytes posted on their social media accounts that only a conspiracy theorist would claim that the virus hadn't been isolated.
Only a conspiracy theorist wouldn't claim that it has not been isolated.
I mean, at this point, that is totally true. Only
if it walks like a
duck
would
say that.
Quacks like a duck. I mean, it definitely quacks for sure.
Yeah. So because at this point, to say that the virus hasn't been isolated, at this point to say the virus hasn't been isolated, you have to assume that thousands of independent labs that are funded from all different kinds of sources all around the world are all conspiring together to produce fake viral sequences. I mean, that you're a conspiracy theorist if you think that that's true.
You'd have to be. You're just sorry. You are right. Exactly. And it doesn't matter if she's now going to meal around with words on purification, as we talked about purification, totally a different step. So doing this is totally inappropriate.
Let's talk about purification.
She will in a second.
Oh, great. Okay.
The use of the term isolation
so
is anything but clear, and many of the angry messages aimed at conspiracy theorists seem to be by those perhaps not understanding how the word isolated was being used. That is why my co-author, Torsten Engelbrecht, made a point of contacting those authors who had claimed to have first identified or isolated the purported virus to see if they had purified samples. We published our findings in the recently released updated version of Virus Mania, and as many of you will know, none of the study authors claimed to have obtained the virus in purified form.
Because You don't have to purify it to isolate it. That is a
Yeah.
different technique.
Yes, so okay, so for people listening, purification, when it comes to virology and biochemistry, that's a very specific procedure where you actually, you know, you actually by the end of it obtain a very specific material in, it's hard to talk about this without using the term and the definition, but you
Right.
obtain it in a pure form. You know, so if I want to purify proteins, I need antibodies that specifically recognize those proteins, and then I have to use those antibodies in a biochemical assay to pull them out of all the nucleic acids, all the lipids, all the other things that are in that cellular sample. And so same with viruses, you can use antibodies, or you can use a technique called it's called ultracentrifugation, or sucrose gradient fractionation.
Sucrose gradient.
Yeah.
Yeah.
And that's the sucrose gradient. I've done sucrose gradient fractionation. It's fun. It's cool, and you end up with a pure sample of what you choose to collect.
Isolate, because based on gravity and density, they separate into the individual fractions, and then you can take out that fraction, you know, with a pipette, and, and then you have your purified sample.
And I have one paper that I always bring up when people say that this virus has not been purified.
Right.
Which, you know, of course, there's I just put the link in the chat.
Appreciate it.
But
yes, I love this paper. This is an amazing paper, by the way. The ultrastructural imaging it's just beyond unbelievable. I mean, you're getting down to amino acid levels. Go ahead.
Yeah. And if you look at if you just see author affiliations.
Okay.
If you just expand that.
Yep.
Yeah, there's a lot of people who worked on it. So
and they're from Max Planck, you know.
Lots of different institutions. They would all have to have conspired for this. I mean, but yeah, if you go to the methods, they talk about how they purify the viral particles using sucrose gradient fractionation.
Right. Right. I'm just gonna pull out here just to admire the level of resolution that they have, where they're looking at individual spikes coming off of the coronavirus. I mean, that's crazy. And then the way that the hinging works, it gets down specifically to the amino acid level. Amino acids, as you've pointed out, are predicted in the genome of the virus. Yeah, I mean, how much more direct evidence could you possibly need?
I don't know. Yeah. So for those who don't know, the genetic code is a string of letters of A's, T's, C's, and G's. And the combination of those letters will code for a string of amino acid proteins, which are denoted by letters as well. And you can read the genetic code, and you can, on paper, translate it to write out exactly what the protein sequence is going to be. And so when they actually see the physical spike protein on the surface of the virus here, the sequence of those amino acids match the genome based on what you'd expect. So
based on what you would expect, based on the angling of it too. So thanks for pointing that out. Sometimes I forget that people have, you know, cursory knowledge, and we have to bring them up to par on things. So yes. So DNA to RNA, RNA to, you know, well, in this case, it's RNA to protein. And then that RNA code predicts the amino acids which form the protein.
Exactly.
That's right here. And then I'm not sure, is supplemental material that they have the isolation?
In the, when they describe, I think if you just search for,
sucrose.
I think you should be able to find
sucrose identification.
Search for that.
Right. Right. And these are incredibly purified.
Yeah, they describe it right there.
Mm-hmm.
Yep.
Yeah, so
So
there's nothing that's gonna. But she will bring up another paper where they're saying like, oh, well, these authors were sloppy, but then she uses that. You know, this paper said the authors were sloppy. They're looking at ultrastructural images not at this level, not at this resolution, and making conclusions that could be ribosomes, that could be vacuoles, that could be other things because their resolution isn't good enough. There's no mistaking this. This is a coronavirus.
No, yeah, it is definitely a coronavirus. But okay, you made me curious now. I want to see what she says about the ultra structures.
Oh yeah. Let's get to it. So, and again, these people they've isolated the bacteria the virus by doing what we told them, what we said they do to isolate it. You know, they plate that out on culture cells. And then they keep doing that until they get a culture that they can contain, which has the virus in it. And that's not super purified. It has cellular debris in it because viruses have to live in cells.
Right. Yeah. And so one last thing. It's frustrating because it's kind of like this perfect storm of confusion that she can sit in, because while isolate does not have a universal definition among virologists and just bacteriologists in general, purify does. Purify, everyone knows what you mean when you say purify something. I mean, that is a very picky, specific thing that scientists, virologists and biochemists do have in their vocabulary. So when asked, did you purify it? Of course, they're going to say, well, no, we didn't purify it because we didn't have to.
We didn't have to. And also they will pick out things like, even in the paper that you said, I showed it to someone and they're like, well, it says that they used like paraffin wax. And it's like, that's not purified. And it's like, no, you have to use stuff. Yes, exactly. It's like, you have to use that in order to, to hold the sample steady in order to take these, ultra micrograph images. But it's like, they'll just move the goalposts. It's like, well, no one has virus particles that are in just pure DDHO or just like dried, I guess is what they want. They will always move the goalposts and say that it's something else or it's some other component that you haven't, it's goalpost moving is rampant. And this is basically the start of goalpost moving.
Yeah.
Now, if we go back to our dictionary definition of isolate, which is to obtain a substance or microorganism in an uncombined or pure state, we can see that people are confused, or at least talking about very different things, because nobody has produced an uncombined or pure state sample. Other investigators were also aware of the inconsistent ways in which isolation was being used. Here in New Zealand, the media reported in mid-2020 that Professor Quinone Matteau of Otago University was able to grow the SARS-CoV-2 virus and isolate its RNA, its genetic material. However, Canadian researcher Christine Massey and New Zealander Michael S. have been submitting freedom of information requests to health institutions around the world. When they made an Official Information Act request to Otago University, they were advised that the university, in fact, had no records of any study describing the actual purification of the alleged virus. Okay, it's time to get some more lessons from Professor Racanello to see if a representative from the orthodoxy can tidy up their mess. He says, an isolate is a virus that we have isolated from an infected host and we have propagated that in culture. You put that sample in cells and culture, which is how we grow viruses.
It just completely undoes her entire framework in the past of saying how you isolate viruses and purify them and why they don't need to be purified at that level. So, but she'll go on and she's going to really abuse Vincent Racanelli's stuff. So, here we go.
And you grow a virus and now you have an isolate. All right. I think we might have hit some major problems here.
Not if you understand virology, you didn't hit any major problems. That's how it works.
Yeah. Okay. Let's see what she says.
He's implying that someone spits in a cup, and if you mix it with a cell culture, hey presto, we've isolated a virus. That doesn't sound like isolation at all, because we're dealing with samples and culture mediums that contain all kinds of substances without any mention of purification. With regards to this, in Virus Mania, we quote Nobel laureate virologist Françoise Barré-Sinoussi as saying, "You have to purify the virus from all this mess." Professor Racaniello goes on to say that most of the time we take this nasopharyngeal swab in the solution, we just do the genome sequence, and we don't actually have a physical isolate virus, and that's very important. Okay, so that's a big concession. He admits many of these so-called isolates are simply detecting genetic sequences from crude clinical specimens. Unfortunately, Professor Racaniello's discipline for terms really seems to fall apart when he goes on to say, this is a phylogenetic tree of 4,000 or so genome sequences of SARS-CoV-2 isolates. But then he goes on to recover from this slip and state that the dots are individual genome sequences which may or may not have an isolate associated with them. But can you see the major problem here? Detecting nucleic acid sequences does not equate to proof of a virus. In fact, the professor said so himself in his 2017 blog. Many laboratories choose to assay the presence of viral genomes by PCR. This is an acceptable technique, as long as the limitations are understood.
Right. So here now she's morphing
Okay. Yeah.
detection of the virus into Vincent Recanelli's blog. Oh, it's bad. I mean, it's real bad.
Yeah,
First off, the variants are sequenced genomically. The whole genome is being sequenced. This blog is talking about. This blog is talking about the mice paper with Zika, how they found it was sexually transmitted, and it was detecting viral sequence past when it was infectious, because the viral sequence will hang around longer than the actual infectious agent, which is known, but it still is indicative, totally indicative, that the virus was there. They literally injected it into the mouse. So yeah, total abuse.
Yeah. So she is like, I mean, she's conflating a lot of things here. Right. I mean, she shows on screen, all that next strain phylogenetic tree.
Mm-hmm.
Yeah. Which totally destroys her argument and then just scurries around it. I mean, that was wow.
Right. Scurries through it.
But then she goes into talking about how PCR positive is not infectious. I mean, well, first of all, that has nothing to do with the next strain thing, because as we talked about, they're sequencing the whole genome. It's not just a PCR positive. And then she goes into this realm of a PCR positive does not mean you have infectious virus. And that's understood generally. But there's, a doctor does not diagnose COVID based on a positive PCR test alone, because you can test positive and not be diseased. So you're not sick, but you are you could be carrying the virus. So something I hope I've made clear in my videos is that if you get a positive PCR test, first of all, the data are going to be reviewed by an analyst and the supervisors of that analyst. So, people complain about CT values, but if your sample has a high CT value, that is outside the range of the standards built into that test, then it's going to be flagged as suspicious.
Suspicious, and rejected.
It would probably be,
rerun.
Actually, if it was outside of the range of the standards, it would be classified as undetermined. So you would have to take another test. But so if it passes these things, it's going to be reported as a positive. And but you don't normally see your CT value, but the analysts and everybody see that. But if you get a positive, it means one of several things. It means you are carrying SARS-CoV-2.
Or you had.
And it means you're carrying SARS-CoV-2 or you had been carrying it, exactly. And within those two scenarios, it could mean that you caught it, you had an asymptomatic case, and you're recovering from it. That's still important to know. You would want to know whether or not you have had COVID before. It could mean that you caught it and you're in the early stages and you don't yet have symptoms, which is really important to know. Or it could mean that you are just currently an asymptomatic carrier, which is important to know. And it could always mean a false positive. Right, yeah. But it could also always mean that it's a false positive, but again, that's really rare to happen. I mean, that's very, very rare.
It's rare. So in context, I have had 13 of those damn nasal pharyngeal texts, which they really go back there. They're not fun.
Yeah.
Just so that I can maintain, teaching and interacting at my college and interacting with people in the public. They've all come back negative on my part. Obviously that's anecdotal because I'm one person, but I'm just saying I've had 13 of these. I've not had a single positive. These people make it believe make the PCR seem like, well, it's 50-50 whether you're going to get a false positive or not. And they also are conflating the low CT value with a false positive. It's like, well, if you have a high CT value because you're getting over it and you're not infectious, then it's a false positive. No, it's a true positive. You have had, and it's exactly with this, you have had exposure, you have had it, you're just not transmissible at this moment. Or you will become transmissible because you just got it. So
yeah, and you can't necessarily tell the difference unless you get a follow-up PCR test. So unless if you get a PCR test and you have a high CT value, which means you have a low viral load, and then you get another test and your CT value goes lower, which means your viral load is increasing. That probably means that the virus is propagating in you. But in the absence, you could spread it. You could be at risk of spreading it, and that's important to know. But in the absence of that follow-up test, just take the PCR test seriously. Just be safe, stay home. I think the recommendation now is to, if you're asymptomatic, to self-isolate for 7 days.
7 days. Yeah.
And then if you don't develop symptoms, you're good. You're fine. But just take it seriously. It's not nearly as inaccurate as so many people are making it out to be. It's crazy to me. I never thought that I would have to. I never thought I'd spend so much time debunking all these myths about PCR. Yeah, it's crazy to me.
Well, it's people like Sam Bailey that is pushing this stuff, and it's resonating. And I think it's a form of concentration, like so many other of the outlets. I mean, you were around for the plandemic.
Yeah.
That one was, what a cluster that was. And I think those people were still around, and they were still looking for an outlet to tell them that this is my life didn't actually have to be disrupted. It's these damn scientists that are just playing games, and it's not. It's a real thing. You could spread it unknowingly to somebody who is susceptible to it. And in the U.S., you're in the U.S., and actually, I've heard Sam Bailey is also in the U.S. We've had 550, probably more, 1,000 people who've died from this.
Oh yeah, it's the number just keeps growing.
Yep. And it will keep growing despite vaccines. And she has a vaccine video that she can't even publish on YouTube, by the way, because it's so against their policies. And if she does it, she's playing a very intrinsic game here where she's trying to draw people over onto another platform. And she knows she's in the wrong, so she's pushing it over on another platform. Yeah. But all right, so just back to this again. She's obfuscating two separate things.
Right.
PCR detection is not infectious virus. This is true, but it's very indicative that the virus was there and present and exposed. Yes. And whole genomic sequencing to produce these variants. And if it was just random mess of of PCR malfunction in these genomes, they wouldn't form into clades like this.
Mm-hmm.
Yep.
Mm-hmm.
So.
Yeah.
Yep. All right.
It detects nucleic acids, not infectious virus. However, even with the professor's stricter definition of isolate, that of a culture mixture, he's still not referring to a purified specimen. So in fact, how has it been determined that any of these 4,000 RNA sequences on the NextStrain website are viral in nature? You may be interested to know that the prize for providing the proof of isolation and genetic content for SARS-CoV-2 has jackpotted up to €1 million.
Why not $1 billion? It could have been trillion, Debunk. We could be the first 100 trillionaires or quadrillionaires.
Wow.
It doesn't matter. These are bogus challenges. They will never. If you go to this page, have you been to this page, by the way? The quote unquote isolate the truth.
I don't know. I haven't been to this specific page. But go on.
There's nothing that you can't fill out a form to like submit it and just be like, no, you're wrong. It's been proven months and months ago, if not a full month ago. Nothing. There's no way to do it. There's no way to get a hold of anybody. It's what's called a bogus claim. They do these for climate change. They do these for vaccinations. They do these for anything that they don't have direct evidence to. They put up a bogus challenge that will never be fulfilled, so that they can always point to it and say, this challenge has never been fulfilled. So must be that the evidence doesn't exist.
Yeah. And I made a video addressing, I forget how much money it was, honestly, because obviously I never got the money. But it was a challenge to demonstrate that COVID is real, essentially.
Right.
And I think they had something like 11 or 12 qualifications. All of them are like, show that it's been isolated, show that it causes disease, show, all these basic things that have been demonstrated over and over again. So I went through it. I made that video, and I actually sent it to the website had an email, so I sent it to that email, and a middleman got back to me and said, I'll send this along, but I don't. It's basically an impossible challenge because we have plenty of evidence that germ theory is false.
And the middleman said that.
Okay.
Okay.
Yeah, I was like, okay. He's like, okay, I'll send it to our experts. Like, okay, well, I'll be curious to see what they say, and I never heard back.
I would be interested to know if Sam Bailey thinks that germ theory is false. And then she slips up on other occasions too, where she says things like, in her PCR video, she said PCR is really good for using as forensic evidence, and then went into this homage about Carrie Mullis, totally ignoring the fact that Carrie Mullis was going to go on the stand to say PCR couldn't be used for forensic evidence. So
really?
Yes.
Yeah. Wow. So Carrie Mullis just like totally abandoned PCR and all of its applications later in life.
Oh yeah, well, he left that company on bad terms, because if you read that UC Berkeley intolerant genius write up by the Alumni Association, he threatened somebody with like a gun, and they were like, "You're fired, dude."
Oh, okay. I didn't know that much. Wow.
Yeah. He was crazy.
Wow.
And you really get his perspective only from his perspective, because he wrote his own books and his own autobiographies.
Right.
Yeah.
Yeah.
But the people around him,
he
was super intolerant. And what really bothers me about Carrie Mullis is that if it wasn't for the scientific team that was at the company that he was working for, PCR would have never came to fruition. It took the team to take that concept and be like, okay, this is how you do it experimentally, because he was so bad at experimental design that he couldn't show that it would actually work. Anyway.
Yeah, he genuinely had a brilliant idea, but science never happens in a vacuum. That's one problem with Nobel laureates as a concept, is usually Nobel laureates are people who run labs.
Yes.
And the way a lab works is there's a there's a PI, or sorry, a principal investigator,
Yeah.
who is basically the faculty professor who runs the lab and may or may not do experiments themselves. But then all the postdocs, PhD students, and undergraduates that might work in the lab, they're physically doing most of the experiments, and they don't get the credit for a Nobel Prize. I'm not saying that every person in the lab deserves the credit, because that's not true. But it just goes to say that more than one person is responsible for the work and the ideas and the process. And that is missed by saying that Kary Mullis is the, inventor of PCR.
One inventor of PCR. And it totally lacks the concept that a process like Sanger sequencing came out a decade before PCR, which uses a forward primer, amplification by DNA polymerase, and even incorporated these the special DNTPs. DNTPs are nucleic acids that are used to string together to make the DNA tree. And for people who don't know, obviously, you know for sure. And they have these special stop ones so that they can.
Sorry, I was going to say DNTPs are like the little Lego blocks that together will form a long piece of genetic material.
Yeah. And so anyway, Sanger sequencing is very interesting, and I think is even far more complicated than PCR actually was, and it predated PCR by like a decade. So, but okay. Bogus challenge. Anyway, let's see it.
Come on, virologists. Surely that must interest you. So has this misuse in terminology happened recently in virology? It would seem no, going by this letter to the editor that appeared in the Lancet in 1978. C.R. Madeley, a virologist in Glasgow, and C.J.K. from the Department of English Language at the University of Glasgow put forward their plea to clarify the language being used in virology. They stated, an isolate may be defined as a microorganism grown in pure culture, and this implies that the organism has been grown, often through several passages, particularly for viruses, and is therefore available for further study, and that as far as can be determined, only one strain of the organism originating from the patient is present. With regards to other purported detection methods that were being used, they said to refer to positive results in these tests as isolates must be incorrect, since they have not been grown, nor can they be said to be free of other organisms. So over 40 years ago, they warned that isolate was a misnomer, but unfortunately it seems their warning was largely ignored. What actually goes on is at best detection through indirect methods or electron microscopy of unpurified specimens. With regards to this, you can watch my video, COVID-19 Behind the PCR Curtain, to see some of the problems with using PCR as a so-called detection test. And a quick word on the images you might see. Not the lurid computer-generated sea mines that the mainstream media love to show you, but electron micrograph images produced by researchers.
Bait and switch here. I'll show you.
Well, that's definitely not an isolate in the strict sense of the word, because it simply shows nanoparticles near a cell wall. Cells in culture grow, degenerate, and die, producing different nano-sized particles from the cell's surface. You may have heard terms such as exosomes or extracellular vesicles describing these particles.
Oh no.
You might not be surprised to hear that these terms have not been used consistently in the literature either. Speaking of poorly defined particles, I just discovered this gem of a publication that appeared in the Lancet last year. It seems that some groups producing these images are calling fraud on other groups' apparent virus images. They complain that the images in these publications show putative virus particles that lack sufficient ultrastructure for an unambiguous identification as virus.
Ouch. Okay, first off, it's not fraud. They're not even calling fraud. They're calling a misunderstanding, which fraud is what she's doing, which is she pulled this bait and switch here with these ultracellular images. I'm just going to mark this spot quick.
Let's see.
So these are the ultracellular images, are clearly coronavirus. They come from a very famous paper. I can't remember which one it is. I have it cited in some of my other videos, but you can see the nuclear membrane, the the bind RNA in the envelope. Sorry, and then you can see the spikes pretty easily.
Yeah.
So then she quotes the Lancet paper, which is down here. And you can see which ones that they're complaining about, and tell me if this is the same.
Okay.
Close up. So here's the paper that they're saying it doesn't have the amount of. Okay, come on. Yes, I'll accept cookies. Okay, that's what they're saying. Doesn't have it. Oh, come on. There's a problem with doing everything live.
Oh man.
Oh, okay. So here's what they're saying: doesn't have the ultra structural images that prove that it's a coronavirus, and they're correct. This isn't at the resolution that you need to show a coronavirus. This is at the resolution to show organelles in a cell.
Yeah. What's the scale bar there?
100 nanometers. Yeah.
Okay.
I don't know if the other one has a scale bar. I think it does on the original image, but I'll have to pull it up. But these are way smaller than 100 nanometers.
Yeah.
Actually, this scale bar is right here. This one has a scale bar.
So yeah, I mean, it's not. Oh, this one has it. Yeah, this one.
30 nanometers.
Yeah. So that's that's cryo-electron microscopy. That's super tiny stuff. And I'm guessing, is this over here? Are they doing TEM or SEM?
It looks like an SEM to me.
SEM. Okay, yeah, they're not going to be able to make a positive ID with SEM.
They can't. I'd have to. It looks like they just call it an EM. I don't know why, but it looks like shuffle caption. Yeah, they don't say if it's TEM or SEM. It's probably TEM. But yeah, you can't get down that far, and they probably didn't have the ability to do it, and they're relying on testing. But this is a bait and switch to say that all the electron microscopy. It says that the ultra cellular components is not visible, and you're right. I don't see, and nobody will see these intricate detail. Actually, I'm just realizing you can't see. They won't see these intricate details of the envelope and the spikes at that level.
Mm-hmm. Yeah.
Yeah.
Yeah. So and so, yeah, I don't know what the rest of that paper was doing, but whenever there are a couple papers that describe isolation of SARS-CoV-2 and include most of them include an electron micrograph image of the virus, and it'll be an image that might look like this, or it might look more like the previous picture. But either way, you know, at best with this picture, you could say that's probably a coronavirus, but you couldn't possibly identify it as SARS-CoV-2.
No.
But what the scientists are sometimes really clever, because we think about multiple ways to demonstrate the same thing. So, you know, in a paper that's describing an isolation of SARS-CoV-2, they'll have sequencing data. They will have clinical data from the patient. They will have possibly other biochemical tests. And then at the end of it, they'll slap on, hey, we took a picture of it too, and it looks like a coronavirus. So
yes,
all of our data are consistent. Boom, 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 boom. We have this long list of consistent data, and the electron micrograph is just a part of it. You know, they're not making a positive ID on the picture alone. And you couldn't do that unless you were doing cryo-electron microscopy, and you could see the amino acids.
Right. Exactly. But anyway, she's whittling around in that to throw all of the electron microscopy out the window, which is a total bait and switch.
I'm convinced she knows exactly what she's doing.
She
knows exactly that she is lying. I mean, it's so sad to see.
She pulled this image from a very specific paper, this one right here up on the screen, which is famous. And there's no way that she could go through this paper and refute the aspects that are actually in this paper. Instead, she pulls up this image and then puts another paper saying that, well, this other paper did ultrastructural imaging, not showing this image that, well, it's difficult to see because the resolution is only at 100 nanometers, not at the 30 nanometers that you need.
Mm-hmm.
Yep.
Yeah.
All right, let's finish this up.
Yeah.
Good to see. So back on topic, why on earth has all this murky terminology not been resolved in what should be a highly scientific field? After all, virologists say they've been isolating viruses for around a century now. One starts to wonder whether they are not all that enthusiastic about clearing up these terms because it shines a spotlight on one of their ticklish spots. Actual direct proof that they are isolating viruses in the true sense of the definition.
I've cut slices so thin, I couldn't even see them.
How'd you know you cut it?
Well, I guess I just assumed.
As you may realise, the repercussions for this are huge, and there will be further videos expanding on this in the future. Before I go, perhaps you'll find a little irony in this quote from Confucius. If language is not correct, then what is said is not what is meant. If what is said is not what is meant, then what must be done remains undone. If this remains undone, morals and art will deteriorate. If justice goes astray, the people will stand about in helpless confusion. Hence, there must be no arbitrariness in what is said. This.
There we go. We've gotten to the end.
Topping it off with a Confucius quote. I guess that kind of summarizes the video. I mean, what more do you need to know? But
no, exactly.
Oh man,
exactly.
all
right. Well,
that hurt.
Yeah, I'm sure you need to de-stress from that.
I guess the last thing I would say is either she is lying, which I think she is,
right,
or she is not an expert in virology and therefore she is confused by the terminology used in virology. However, virologists are not confused by their own terminology.
No, they're not. They know what an isolate is.
I mean,
they can identify what a variant is, and they use these accordingly and have isolated the isolates that can be, passed between different labs for testing and sequenced repeatedly and tested. So,
and then if desired, purified.
And if desired, purified for micrograph imaging. But okay, well, Debunk, it was great talking to you. This is the problem: a 14-minute video, stopping every 13 seconds to say, yeah.
Well, since this was super fun, I honestly hope we do it again.
Yeah.
Wow.
I got some other guys we can bring in on this mix.
It's fun going over. Yeah, it's fun going over a video like this, especially about a video that I'm not going to formally make a video on. So this is fun. We should do more often.
Cool. All right, we'll do it again. All right, everyone out there, thanks guys. I appreciate it. Check it out. If you really want to and you want to get into it, Vincent Racanelli's video, that's the one to go to. And he's going to say things that are totally counter to what she's saying. And you'll get much better information off of that. But as of them, we bid you adieu and good luck out there.
See ya.
Yeah.